students, in today's lecture, we are going to study different categories of the enzymes. We basically categorize enzymes into two main categories, into two main divisions. First one are the plasma, functional plasma enzymes. The second one is the non-functional plasma enzymes. As the name represents the functional plasma enzymes. These are those enzymes which are specifically designed to perform their particular function within the plasma. These are actually the small group of the enzymes that are secreted by the cells into the blood, into the plasma. The examples include the liver enzymes, that are the zymogens, which are basically secreted in the blood to perform the function of the blood coagulation. Now moving towards the second category, that are the non-functional plasma enzymes. So these are the enzymes which are designed to perform their particular function within the cell. Well, what happens that during the cell damage or during the normal cell turnover, there is release of these enzymes into the plasma or into the blood. So these are actually the active intracellular enzymes, but after certain cell damage, they are released into the plasma. And now in the plasma, these enzymes don't have any physiological role. So what are the examples of the functional plasma enzymes? As I have already discussed, first example include anticoagulants. Then we have certain other enzymes, as we have already discussed in our pharmacology class, that we have acetylcholinesterases, we have butyrylcholinesterases, we have pseudocholinesterases, which perform their particular functions regarding the acetylcholine into the plasma. Now, what are the examples of the intracellular or the non-functional plasma enzymes? These include different liver enzymes, for example, ALT, AST. So now going into the differences between these two enzymes. There are certain differences between these two enzymes. But before going into detail of these differences, look at this diagram. This is representing that we have a normal cell that is releasing its enzymatic content, or that is releasing its enzyme into the plasma at a certain or the controlled rate. I was discussing normal turnover. Now what is turnover? Turnover is the continuous process. It's a normal process during which there is breakdown of the damaged cell and formation of the new cells. But during this process, there is release of the cellular content into the plasma, and one major component of these cells are the enzymes, and these are also released in the plasma, but at a certain and a controlled rate. While in case of cell necrosis, cell damage or the tissue damage, there is release of greater amount of these enzymes into the plasma. Now coming towards the differences of the functional plasma enzymes and the non-functional plasma enzymes. Now, concentration in the plasma. So functional plasma enzymes are present in plasma in higher concentration in comparison to the tissues, because they were naturally designed to perform their particular role within the plasma. While in case of non-functional plasma enzymes, these are present in the tissue or in the cell in higher amount as compared to that in the plasma. They are only released in the plasma whenever there is a cell damage, or they are released in the plasma during the cell turnover. Now, these functional plasma enzymes have specific roles within the plasma, well-designed roles, while these non-functional plasma enzymes don't have any known role in the plasma. Substrate for these functional plasma enzymes are present within the plasma, while the substrate for these non-functional enzymes are absent in the plasma or in the blood. The site for the synthesis of these functional plasma enzymes is the liver, as I have discussed zymogens. But the location of these non-functional plasma enzymes are the cells or the tissues, for example, liver, heart, brain, and the skeletal muscles. Now, what is the effect of diseases on the level of these enzymes? We know that the region of the functional plasma enzymes is the liver. So, if there is liver damage, there will be increased amount of these functional plasma enzymes in the plasma. While in case of tissue or the cell damage, there will be increased enzymatic content, there will be increased non-functional plasma enzymes in the plasma. Now, what are the examples? The examples of the functional plasma enzymes include clotting factors, for example, prothrombin, lipoprotein lipase, and the pseudocholinesterases. While other examples are acetylcholinesterase, butyrylcholinesterase. Now, what are the examples of the non-functional plasma enzyme? These examples include ALT, AST, CK, that is creatinine kinase, and LDH, alkaline phosphate, acid phosphate, and amylase. So these are the differences between the functional and the non-functional plasma enzymes.